Hello everyone, welcome to another video by Totality Education. Today we are going to discuss in detail about meristematic tissues and permanent tissues under the topic tissues. So this is part 2 of tissues. Part 1 is linked down in the description and in the top right corner. So please check that out and let's get started with today's video. So let's start off with meristematic tissues. What are meristematic tissues? So we've discussed that in brief in our previous video. This time let's discuss meristematic and permanent tissues in detail. So nature. Cells of meristematic tissues divide continuously and help in increasing the length and girth of the plant. These cells show the following characteristics. So the characteristics of meristematic tissue are on your screen right now. So let's read them one by one. The cells of meristematic tissue are similar in structure and have a thin cellulose walls. The meristematic cells may be spherical, oval, polygonal or rectangular in shape. The meristematic cells are compactly arranged and do not contain any intercellular space between them. So each meristematic cell contains dense or abundant cytoplasm, a single large nucleus. The meristematic cells contain few vacuoles or no vacuoles at all. So as you can see here in this image, that these are the cells of the meristematic tissue. We all know that a group of cells makes up a tissue. So over here you can see that this is a meristematic tissue. As we have discussed, they are either spherical, oval or polygonal or rectangular in shape. Here we have hexagonal meristematic cells. So you can see that they are all in almost the same shape and they have a single prominent nucleus in each of them, and you cannot see much vacuoles anywhere, and they have a dense cytoplasm. So there's like cytoplasm everywhere and they are compactly arranged, so they don't have that much intercellular space. So that's what we have learned. The meristematic cells are compactly arranged and do not contain any intercellular space between them. Even if I zoom in really close, I cannot find any intercellular space. So that's the meristematic tissue. Now let's discuss about the occurrence of meristematic tissues. So we need to go to this image over here. So this is the shoot tip of a plant. So the occurrence of meristematic tissues are growth tissues and are found in the growing regions of the plant. According to their position in plant, meristematic tissues are apical, lateral and intercalary. So when we classify meristematic tissues, we get apical meristem, intercalary meristem and lateral meristem. So let's discuss each of them in detail. So apical meristem, what I'm gonna say right now is on your screen about apical meristem. These apical meristems are situated at the growing tip of stems and roots, that is, at shoot apex and root apex. Apical meristems are also found at apices of the leaf. So this is the apical meristem, as you can see in the diagram. This diagram is there even in your NCERT textbook. So it's at the tip of the plant. If you happen to cut off this apical meristem, the plant may not grow. So this is the apical meristem. So this helps the plant to grow taller. Now let's discuss about lateral meristem. These are found beneath the bark, also called cork cambium, and in vascular bundles of dicot roots and stems called vascular cambium. They occur in thin layers. Cambium is the region which is responsible for growth in thickness. So in thickness. And then we have intercalary meristems. So you can see the lateral meristems are somewhere over here at the bottom. These are the lateral meristems, and the intercalary meristems are over here. And okay, so intercalary meristems, they are located at the base of leaves or internode. Example, stems of grasses and other monocots such as tissues or also occur below the nodes. The functions of meristematic tissues I've shown it in the previous video. If you want, you can check that out, and again, it's in the description and on the top right corner. I'll just read out the functions for now. Meristematic tissue acts as a parent tissue from which other tissues develop. These tissues take part in growth by formation of new cells, which with the help of meristematic tissues, plants continue to produce new leaves, branches of stem and root, flowers, fruits, and root hairs. The place of injury in plants is healed up by the formation of new cells by meristems. The plant shoots lodged or bent by wind are made to grow upright by the activity of intercalary meristem. Different types of meristem have the following functions. So apical meristem. So these functions will be on your screen right now of apical, lateral, and intercalary. It brings about an elongation. Apical meristem, it brings about an elongation of root and stem. It results in the increase in the height of plant, which is called the primary growth. Lateral meristem, it causes the organ, stem or root to increase in diameter and girth. This is called secondary growth. For example, cork cambium or phelogen produces a protective cork or on the outside of secondary cortex tissue inside. Intercalary meristem, it produces an increase in the length of an organ such as leaves and internodes. So there's an activity which you could do to observe the growth of meristematic tissues. So this is there even in the NCERT textbook. So you take a beaker and you put an onion inside, and after a few days you can see that the roots of onions have grown. This is because of the intercalary apical meristems. So you can see over here the apical meristems. So these grow and make it a little longer. Now let's move on to permanent tissues, and then we will discuss about revise about meristematic tissue and permanent tissue, the differences between them, even though we have already done that before. Now let's discuss about permanent tissues. What happens to cells formed by meristematic tissues? Cells derived from division of meristematic tissue take up specific role and lose their ability to divide. They thus form a type of permanent tissue. The developmental process by which cells derived from meristematic tissue take up permanent shape, size, and function is called differentiation or cell differentiation. We have defined this in our previous video. In this way, cells of meristematic tissue differentiate to form cells of permanent tissue. Different types of permanent tissues are formed due to differences in their specialization. Permanent tissues may be simple or complex. So that's the two types of permanent tissue, simple and complex. Their cells may be dividing or dead, thin-walled or thick-walled. Thickening may be regular or irregular. So we have two types of permanent tissue: simple permanent tissue and complex permanent tissue. So let me write that down here. So we have simple permanent tissue and complex permanent tissue. Simple permanent tissue, and the other one is complex permanent tissue. Complex permanent tissue. So these are the two types of tissues of permanent tissues, and these can again be classified. So simple permanent tissue will be classified into parenchyma, collenchyma, and sclerenchyma. These names are a little difficult to remember, but so we have parenchyma. Where did that go? So we have parenchyma. Please make sure the spelling is right. It's Parenchyma, parenchyma. And then we have collenchyma. Please be careful of the spelling. It's not Chllonchyma, it's Collenchyma. That's the correct spelling. Collenchyma. And then we have sclerenchyma. Let me just check the spelling of that. Sclerenchyma. Sclerenchyma. So these are the three types of simple permanent tissue: parenchyma, collenchyma, and sclerenchyma. We will be discussing about them in detail a little later. And then we have something called as complex permanent tissue. So when we divide that, we get xylem and phloem. I'm sure we have learned that when we are in our lower grades. We have xylem and phloem. Xylem, XYLEM, xylem. And then we have phloem, PHLOEM, phloem. We have xylem and phloem under complex permanent tissues. So these are types of permanent tissues: simple permanent tissue, complex permanent tissue. Under simple permanent tissue we have parenchyma, collenchyma, sclerenchyma, and under complex permanent tissue we have xylem and phloem. So xylem are again divided into four types: tracheids, vessels or tracheae, and we have xylem parenchyma, xylem sclerenchyma. And then even phloem is also divided and they're divided into sieve tubes, companion cells, phloem parenchyma, and phloem fibers. So we'll soon discuss in detail about all of them. And also after permanent tissues we are going to discuss about protective tissues. So now let's discuss about parenchyma. So the nature of parenchyma is that it forms the bulk of the plant body. Parenchyma cells are living and possess the power of division. The cells are rounded or isodiametric, that is equally expanded on all sides. So isodiametric means that it's equally expanding on all sides. So now the parenchymatous cells are oval, round or polygonal or elongated in shape. The cell wall is thin and encloses a dense cytoplasm which contains a small nucleus and surrounds a large central vacuole. In other words, parenchyma cells have living protoplasm, intercellular space, and are abundant. So here we have said that they possess the power of division. That means parenchyma cells are divide continuously. So that's why the cytoplasm is dense and contains small nucleus and surrounds by a large vacuole, etc. So occurrence: the parenchyma is widely distributed in plant bodies such as stem, roots, leaves, flowers, and fruits. Thus the parenchyma tissue is found in soft parts of the plant body such as cortex of roots, ground tissues in stems, and mesophyll of leaves. It is also distributed in pith, medullary, rays, and packing tissue in xylem and phloem. Functions: parenchyma serves as a packing tissue to fill the spaces between other tissues and maintain the shape and firmness of plant due to its turgid cells. Due to turgidity property, parenchyma acts as a primary support to the stem of herbaceous plants. The main function of parenchyma is to store and assimilate food. Parenchyma serves as food storage tissue. Starch present in amyloplasts in parenchyma of cortex of potato tuber. Transport of materials occurs through cells and or cell wall of parenchyma cells. Parenchyma cells are metabolically active. Their intercellular air spaces allow gaseous exchange. Parenchymatous tissue stores waste products of plants such as tannin, gum, crystals, resins, or inorganic wastes, etc. If chloroplast is present, the parenchyma tissue is called chlorenchyma. So I'll type the spelling over here in red color. Chlorenchyma. So chlorenchyma. This is the spelling of chlorenchyma. It's what makes the parenchyma green in color. And then we have, and the next property, in hydrophytes such as water hyacinth, hydrilla, large air cavities are present in parenchyma to give buoyancy to the plants. Such a type of parenchyma is called aerenchyma. If you want to know what buoyancy is, comment down below. I'll make a video on it. So we have aerenchyma. So I'll type the spelling of aerenchyma here. Aerencyma, aerenchyma. So the spellings of chlorenchyma and aerenchyma are over here. In xerophytes, examples, succulents, parenchyma acts as a water storage tissue. So from this, what we can understand is that mainly parenchyma is mainly for the storage, and you can see here in this image that parenchyma has a lot of intercellular space over here. So that's given intercellular space, and it has a large central vacuole because mainly it's for storage, right? Hence it has a large vacuole. And then we have a layer of cytoplasm. Cytoplasm is there in most of the cells, in every cell. It's there in every cell, like nucleus, cytoplasm, and then a huge vacuole. And then we have a prominent nucleus. We have a prominent nucleus over here. So yeah, and it has a primary cell wall. So that's about parenchyma. Over here you can see aerenchyma in a cell, in a hydrilla stem. Over here, these little ones over here are the aerenchyma. These little ones over here are the aerenchyma over there, and the rest we don't need right now. We'll learn about protective tissues later, in the later part of this video. For now, this is it. And after parenchyma, now we're going to discuss about chlorenchyma. Now let's talk about chlorenchyma. So the nature of chlorenchyma is that the tissue also contains living cells. It is known it shows many features of parenchyma but is characterized by the deposition of extracellulose and pectin in the corners of the cells. So you can see in this image here what I just said. See over here you can see that it has pectin in the corners. So chlorenchyma cells are elongated in shape and often contain a few chloroplasts. So in chlorenchyma, intercellular spaces are generally absent. They're generally absent, but they are, very scarcely present. Occurrence: the cells of chlorenchyma are located below the epidermis. So what is an epidermis? We will discuss about it later. And to be specific, it is a hypodermis of dictyloledon stem and petiole. These cells also occur in the midribs of dicot leaves. Chlorenchyma is absent in monocot stems, roots, and leaves. Functions: chlorenchyma is a mechanical tissue in young dicotous systems and provides mechanical support and elasticity. Thus, chlorenchyma provides tensile strength with flexibility to those organs which is found. It allows easy bending in various parts of plant without actually breaking it. When cells of chlorenchyma contain some chloroplasts, they manufacture sugar and starch. Differences between parenchyma and chlorenchyma. So parenchyma, the cell tissue of The tissue consists of thin-walled living cells. It is distributed in almost all the cells of all the parts of the plant body. The living cells of parenchyma assimilate and store food. They also store waste products. Collenchyma. The tissue consists of cells having localized thickening in their cell walls. It occurs mostly in the aerial parts of the plants and is restricted to the outer layers. Collenchyma is the chief mechanical tissue in parts of young plant, particularly in the dichotous stems. Now let's move on to what is sclerenchyma. Sclerenchyma cells are dead cells, and they are devoid of protoplasm. They are dead. Okay, so rest of them, which were parenchyma and collenchyma, they are alive, but sclerenchyma is dead. The cell walls of sclerenchyma are generally thickened due to lignin. Such cell walls are lignified. Okay, so I'll show you. I'll tell you the spelling of lignin. Please make sure not to make a spelling mistake. It's Liginin. There's no I after G. It's Lignin. 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 Such cell walls are lignified. So the spelling of that is lignin. No, is lignified. F-I-E-D. Lignified. 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 Due to cells of sclerenchyma, due to the excessive thickening of sclerenchyma cell wall, the cell cavity or lumen becomes nearly absent. So becomes nearly absent. The cells of sclerenchyma are closely packed without intercellular spaces. Thus, these cells are fitted together like tiles in a mosaic floor. A conspicuous middle lamella exists between two sclerenchymatous cells. Middle lamella is a thin layer of cementing substance containing pectin, lignin, and protein. It occurs between cell walls of two adjacent plant cells. Cells of sclerenchyma are of two types: fibers and sclerids. Fibers consist of very long, narrow, thick, and lignified cells. The length of sclerenchymatous cells varies from 1 mm to 550 mm in different plants. Fibers are usually pointed at both ends and are clustered into strands and look like polygonal transverse section. In contrast to fibers, sclerids, also known as grit cells or stone cells, are irregular shaped. Examples: sclerids may be spherical, oval, cylinder, T-shaped, dumbbell shaped, or stellate. They are dead and develop in various parts of plants such as cortex, pith, phloem, hard seeds, etc. Often oblique thin areas are found in thick walls of both fibers and sclerids. These are called piths. Now let's talk about the occurrence of sclerenchyma. The sclerenchyma occurs in abundance either in patches or definite layers. They are found in stems, roots, veins of leaves, hard coverings of seeds and nuts. Sclerids form the gritty part of most of the ripe fruits and contribute hardness to the seed coat and nutshells. Husk of coconut is made of sclerenchymatous tissue. It is present in mesocarp of fruit of coconut and yield coir, a well-known fiber used for making mats, cordage, brushes, etc. You can see the tissue of coconut fruit on your screen. It's the right picture. Differences between sclerenchyma fibers and sclerids. Sclerenchyma fibers, they are elongated, spindle-shaped, thick-walled dead cells. They are arranged in bundles, nets and cylinders. They do not form covering of any plant origin. They provide mechanical strength. Sclerids. They are broad, thick-walled dead cells. They occur singularly or in small groups. They form hard covering of nuts and seeds. They provide stiffness. Functions. Sclerenchyma is mainly mechanical and protective in function. It gives strength, rigidity, flexibility, and elasticity to the plant body and thus enables it to withstand various strains. So if wind is blown on a plant tree, the sclerenchyma helps it to get upright. So differences between collenchyma and sclerenchyma. Collenchyma, it consists of living cells. Its cells contain cytoplasm. Its cell walls are cellulosic. The thickening of cell wall is not uniform. Lumen of cell is wide. It provides mechanical support and elasticity to the plant body. Sclerenchyma, it consists of dead cells. Its cells are empty. Its cell walls are lignified. Cell wall thickening is non-uniform. Lumen of cell is narrow. It is chiefly mechanical tissue. Next, we will talk about the xylem and phloem, that is the complex permanent tissue, and then we will go for the protective tissues. Now let's talk about complex permanent tissues. The complex tissues consist of more than one type of cells having a common origin. These cells coordinate to perform a common function. Complex tissues transport water, minerals, salts, and food material to various parts of the plant body. Complex tissues are of the following types: xylem or wood, phloem or bast. So it's the two names of xylem and phloem. Xylem is also called wood and phloem is also called bast. Xylem and phloem are both conducting tissues and are known as vascular tissues. Together, both of them constitute vascular bundles. So let's start off with xylem. So the nature of xylem. Xylem is a vascular mechanical tissue. In other words, it is a conducting tissue. Xylem is composed of cells of 4 different types. So we have already discussed about these 4 different types. Tracheids, vessels or tracheae, tracheids, vessels, xylem parenchyma, xylem sclerenchyma. Except for xylem parenchyma, all other xylem elements are dead. That means that xylem parenchyma alone is alive, and the rest, tracheids, vessels, and xylem sclerenchyma are all dead. So and bounded by thick lignified walls. So other than xylem parenchyma, rest of them have thick lignified walls. All these 4 types of cells of xylem, most important cells are vessels. Vessels are shorter and wider than tracheids. Types of cells of xylem than tracheids. Vessels are very long tube-like structures formed by narrow of cells placed end to end. The transverse walls between the vessel elements are partially or completely dissolved to form continuous channels or water pipes. Tracheids are elongated cells with tapering ends. They also conduct water. Since tracheids do not have open ends like vessels, so the water has to pass from the cell to cell via the pits. Xylem parenchyma stores food and helps in lateral conduction of water. Wherever this parenchyma, it basically means that it stores something. So over here in xylem parenchyma stores food and helps in lateral conduction of water. So let's discuss about the difference between tracheids and vessels. Tracheids, they are single-celled. The end walls remain intact. The walls of tracheids are very thick and narrow lumen. Vessels, they are made up of a row of cells and means transverse and equals transverse. Walls get dissolved and become perforated. The walls of vessels are less thick and they have a wider lumen. Functions of xylem. The main function of xylem is to carry water and minerals upward from roots to different parts of shoots. So basically leaves. We've already learned these in our lower grades about xylem. Since walls of tracheids, vessels, and sclerenchyma of xylem are lignified, they have mechanical strength to the plant body. Now let's talk about phloem, and then we'll discuss about the difference between xylem and phloem. So now let's discuss about phloem. The nature. Like xylem, phloem contains tubes but performs no mechanical function. Phloem is composed of following four elements of cells. Sieve tubes, companion cells, phloem parenchyma, and phloem fibers. Except for phloem fibers, phloem cells are all living cells. So this means that phloem fibers are dead and sieve tubes, companion cells, phloem parenchyma are all living. Sieve tubes. Sieve tubes are slender tube-like structures composed of elongated thin-walled cells placed end to end. Their end walls are perforated by numerous pores and are called sieve plates. Walls of sieve tubes are perforated. The nucleus of each sieve cell degenerates at maturity. However, cytoplasm persists in the mature cell. Thus, nuclei are absent in the mature sieve tube elements. The cytoplasm of one sieve tube element is continuous with those of other sieve elements above and below due to cytoplasmic connections passing through the pores of sieve plate. Now, companion cells. Generally associated with the sieve tube is a small thin-walled cell containing a dense and very active cytoplasm and large elongated nucleus. It is called companion cell and it is connected to the sieve tube with numerous plasmodesmata. Phloem parenchyma. These thin-walled living cells of parenchyma or phloem, they have two functions: storage and low lateral conduction of food. Phloem fibers or blast fibers. These are thick-walled, elongated, spindle-shaped dead cells which possess narrow lumen. They provide mechanical strength to the tissue. Bast fibers obtained from some plants such as jute, hemp, flax are commercial for economic value. Now, let's discuss about the functions of phloem. Phloem transports or conducts photosynthetically prepared food materials from the leaves to the storage organs and later from storage organs to growing regions of the plant body. So this is about phloem. Now difference between xylem and phloem. Xylem, it conducts water and minerals. Conduction is mostly unidirectional, that is from roots to apical parts of the plant. Conducting channels or trachary elements are tracheids and vessels. Components of xylem include tracheids, vessels, xylem parenchyma, and xylem fibers. Three of the four elements of xylem are dead. Only xylem parenchyma is living. In addition to conduction, xylem provides mechanical strength to the plants. Phloem, it conducts organic solutes or food materials. In it, conduction may be bidirectional, that is, from leaves to storage organs or growing parts from storage organs of growing plants. Phloem parenchyma and phloem fibers. Conducting channels are sieve tubes. Components of phloem include sieve tubes, companion cells, phloem parenchyma, and phloem fibers. 3 of 4 elements are living. Only phloem fibers are dead. Phloem performs no mechanical function for the plants. So these are the difference between xylem and phloem. Now we will discuss about the protective tissue, which is the epidermis and the cork. So let's talk about protective tissues. Protective tissues include epidermis and cork. Cork can also be called as phloem. Epidermis. So let's talk about epidermis now. Epidermis. The epidermis is usually present in the outermost layer of the plant body, such as leaves, flowers, stem, and roots. Epidermis is one cell thick and is covered with cuticle. Cuticle is a waterproof layer of waxy substance called cutin, which is secreted by epidermal cells. Cuticle possesses variable thickness in plants. For instance, it is more thicker in xerophytic or desert plants. Cells of epidermis are elongated and flattened and do not contain any intercellular space between them, as you can see in the picture. Their inner contents are similar to parenchyma cells. The main function of epidermis is to protect the plant from desiccation and infection. In fact, cuticle of epidermis helps to reduce water loss by evaporation from plant surface, and also helps in preventing entry of pathogen. Do we know what the water loss by evaporation from plant surface through stomata is? Of course, it's called transpiration. We've learnt it. Now, the aerial surfaces of many plants bear cutinized hair over their epidermis. So you can see that in the A over here, the top left in the picture. They are called trichomes. They reduce the rate of transpiration. The seeds of cotton contain numerous long unicellular hairs which form the husk of cotton. In roots, the younger parts are covered with uncutinized layer of epidermis called epiblema. Some of the epiblema cells give rise to tubular outgrowths called root hairs. These long unicellular root hairs increase the absorptive surface area of the root. They pass into soil interspaces to absorb water and minerals. Now let's talk about cork or phloem. Now we have cork. As plants grow older, the outer protective tissue undergoes certain changes. A strip of secondary meristem called phallogen or cork cambium replaces epidermis of stem. Cork cambium is a simple tissue having only one type of cells. The cells of cork cambium are rectangular and their protoplasts are vacuoled and contained in tannins and chloroplasts. Cork cambium gives off new cells on its both sides, thus forming cork or phloem. On the outer side, the secondary cortex or phloderm on the inner side. The layer of cells which is cut by cork cambium on the outer side ultimately becomes several layered thick cork of trees. Cells of cork are dead and compactly arranged without intercellular spaces. The walls of cork cells are heavily thickened with an organic substance called suberin deposits. Suberin makes these cells impermeable to water and gases. The cork cells do not contain protoplasm but are filled with resin or tannins. In case of an onion bulb too, the skin of the cell walls become thick and waterproof due to addition of suberin. Cork is protective in function. Cork cells prevent desiccation, infection and mechanical injury. Cork is light and does not catch fire easily. Due to these properties, cork is used as insulators, shock absorbers, linoleum and sports goods. Commercial cork is obtained from the stem surface of cork oak tree found in southern Europe and North Africa. So that's about cork. Other than that, thank you so much for watching. Like, share and subscribe. And I've noticed that my YouTube channel is not growing much nowadays, so I want you all to subscribe. And other than that, nothing else. Please subscribe. I'm asking you again and again, please subscribe, watch my videos, and like, share, subscribe, everything. Well, thank you so much.